Okay, so we are now moving to chapter 5, which I'm not a huge fan, honestly, of these. I made them, but of these glue ends. So they're half pages, so each one is its own thing, and you're going to have to really pay attention with me because I don't think that makes a lot of sense the way that they're copied. But the one we're going to work with is this left side of the front page, so I'm going to just cut it out. I guess I don't know what I'm going to do because I have to keep up with the other half. The person who copied these, me, could have done a better job. So anyway, this is now loose, and I'm going to keep up with it because proteins are a whole different thing. So we're going to have, let's see, we'll have a video on what are macromolecules, the different classes, and what do we do with them. This is so far past what you do in biology as a freshman. You just need to hang on. I mean, this is true biochemistry, but it's not awful, but it's definitely not what you've done before. So then we'll break out and we'll have all 4 types. So then we'll do proteins and carbohydrates and lipids and nucleic acids. So let's start off with just what is the deal with this. So I know y'all done like, I don't know, vomit lab or all kinds of stuff, but here's what I'm going to do in my notes. I'm going to do like we did for the functional groups, and I'm going to put this strip here, and then I'm going to take notes all the way across. That's what I plan on doing, because there's quite a bunch. So biomolecules, most of the time what you're going to see is we're going to call them macromolecules. And the reason for that is these are large molecules made up of individual smaller pieces. So macromolecules, we can go ahead and we can list what we're talking about. So a protein is going to be a large macromolecule. It's going to be a molecule that's made up of smaller subunits that we can actually classify. That's kind of the definition of what a macromolecule is. It's a large polymer made up of smaller subunits, and the subunits aren't exactly the same, kind of like Legos aren't exactly the same, but they're pieces that are assembled into this protein. So then we've got carbohydrates. And we have lipids. Lipids are kind of a little odd. And then nucleic acids. Each one of these will have its own video. So, when you talk about a biomolecule or a macromolecule, let's just remind ourselves what it's going to be. You're going to have a polymer, and a polymer just means part, piece. So this is made up of many pieces, and this is honestly the best way to draw it. So you'd have almost like beads on a string. Each one of these little pieces is going to symbolize a monomer. So this is the single piece. That's what mono means. So each one of these is a monomer. And they're created by covalently bonding. Not a big surprise to you, I'm sure, because we've talked about covalent bonds non-stop here lately. So you're going to have a covalent bond between each monomer. So something silly to think about is this. If you have look at that little piece. 1, 2, 3, 4, 5, 6, 7, 8. That's not really a polymer, but we're going to go with it. An 8 monomer put together into a polymer. How many bonds would you have? it's going to always be N minus 1, because it's going to be between 1 and 2, 2 and 3, 3 and 4, 4 and 5, 5 and 6, 6 and 7, 7 and 8. So it's always going to be covalent bond. You would have N, which is the number of monomers, minus 1. So N minus 1. So in this case, we'd have 7. If we had 200, it'd be 199. If you had 1,000, it'd be 999. It's always going to be 1 less than the number of monomers that you have. 1,000 is much more realistic than 8. Nobody wants to draw 1,000. So you're going to have that many covalent bonds that where you took a monomer and a monomer, and they were separate. And then you're going to go through a process to create that covalent bond. There is an enzyme, like I've talked to you about, like a little machine, and it's going to come over here, and it's going to have a way of these two sitting in its pocket. So this would be the enzyme. And it's going to bring these two monomers so you've got two monomers. The enzyme's going to bring them together, and the enzyme's job is to put that covalent bond between it. So when they're pieced together, that's called synthesis. It makes sense. Synthesis is building. So this is that process. Two monomers create the covalent bond between them through the process of, so this is a process called dehydration synthesis. And there's a reason why. So synthesis means to put together. Dehydration means by removing a water. I mean, if you're dehydrated, you're low on water. You know exactly what dehydrate means. So don't make this complicated. Don't make it out like, I don't know what that's saying. Well, sure you do. You know what dehydrate is. Dehydrate is to remove water. Synthesis is to put together. So you're going to remove one water molecule. So what they'll usually show is this bond has been made, and they'll have an arrow, and they'll show a water has been removed. So we'll get into a little bit more detail with it, but what tends to happen is on one side of one monomer is a hydrogen. On one side of the next monomer, the opposite side, is a hydroxyl. And this enzyme, so this would be the enzyme, can put those two together into water, and when it does that, it puts the monomers together. So it's going to take a hydrogen off of one and a hydroxyl off of the other. That together gives you water, and the removal of that provides this covalent bond. So I told you, covalent bonds are cool. They're really sturdy, they're stable, unless you purposefully break them or make them, they don't just go away. Like they're not something that's fleeting, like a hydrogen bond. They're fairly permanent unless an enzyme undoes them. So there's an enzyme that puts the monomers together, and the process is called dehydration synthesis because you put it together by removing water. But you have to take them apart too. We make big molecules and then we break big molecules. So hydrolysis, lysis, if you lyse something, that means you cut it. To cut, hydro means water. So you're going to cut with water. So what that would be is, here's your dimer, because there's two monomers put together through dehydration synthesis with a covalent bond. If we add water back, just reverse of this, then you can have your monomers. So you're going to let that hydrogen go back and the hydroxyl go back, and you now have your monomers separated. So this enzyme is going to work here on the covalent bond, and it's going to break it. So it's going to work on the covalent bond. It'll cause that bond to be broken, and it's broken by putting back the water that was removed before in the form of a hydrogen and a hydroxyl. That's kind of it for the beginning. So what you really need to get into your head is one more other thing. So I'm going to come over here and write it. I hope this video works. My computer is way delayed. Like I can watch my hand and it's moving different. It's okay. Let's think about the world. So we're going to go real big. Hello, universe. Here's the universe. We're thinking about the universe. In the scope of the world, you've got disorder and order. So everything moves toward disorder. So that's just something to think about. So everything, which means all living processes, everything moves toward disorder. Decide that you're not going to pick up your room for 6 months and see what happens. That's what I'm talking about. Everything naturally moves toward disorder. Disorder is the same exact thing as entropy. And we talk about this a lot. Entropy is a state of, or a measure of, disorder. High entropy, high disorder. You only move from disorder to order by adding energy in a system. So if you want to move from disorder to order, you have to add energy. You have to pick up your crap, right, in your room. That takes energy because you're actually putting things together that belong together, and you're reducing the number of pieces. So I always make the joke, and this was in your video, and I went and found a picture. So if you get a Lego bin and you dump it all out on the floor, that's high entropy. If you build the Death Star from it, that's low entropy. So here's why I'm saying this. In a biological system, if you have lots of monomers, the building blocks, the pieces, that's going to be high entropy. Like a bunch of Legos. If you put the monomers into polymers, that takes energy. So if you take monomers and you put them together and assemble them into polymers, that has to it costs energy. You have to add energy to the system. And it's going to be in the form of those covalent bonds that you put those pieces together with. And polymers have low entropy because they're ordered. So biological systems have to balance the It's not easy to work in a mess. Think about that. High entropy is a difficult biological system to function within. But how much energy am I willing to spend to not have a bunch of mess and to lower the entropy? So this is a balance within a biological system. So there's always going to be two processes going on. One is a building process. So you're going to go from monomer to polymer. This is building. That is called anabolism. It would be an anabolic process. Now, one way to remember that is, I don't know if you've ever heard, but people talk about anabolic steroids, and they think that's like a type. It's not a type. Anabolic means you build. So they're steroids that are meant to build you up. So anabolism is building, the making. If you go from the polymer to the monomer, which is the breaking, that's called catabolism. And the easiest way to remember that is cats break things. Because there's only two, and just like degrees of freedom, if you know the first one, then you know the second one. So if I'm thinking, I don't remember which is anabolism, catabolism, which one's break and making. Oh, cats break things. That means the other one is building. So anabolic, you're building. This requires energy. What in the world? Energy. Anything that requires energy, these are the words down here that we're going to remember. Building molecules is anabolism. Anything that requires energy is called endergonic. It's going to take energy to make that work. Going from a polymer to a monomer, where you break things, is catabolism. This releases those bond energies, so releases energy, and this is called exergonic. So the words we need to actually pay attention to: anabolic, catabolic, endergonic, exergonic. So that when we see those words, we know. Now, let's add one more little piece of information on here. When you become a polymer, you have low entropy. When you have monomers, you have high entropy. So if we did not put any energy into the system of the universe, things would die. They would break down. We would be having pieces of things. So you would move toward disorder. That's what they're talking about with everything moves toward disorder. So in order to find things, like if you find an ordered system, energy was required to order that system. So we need to talk about the different types of polymers that biological systems make. Those are macromolecules, right here. And each one has its own name for the monomer, and each one serves a different function in a living biological system. So we'll talk about what the functions are, what the smallest pieces are, and then we name the bonds. So yes, they're all covalent, but you wouldn't like if I was like, hey, Student, get over here. You have a name. So they name the bond, the covalent bond, that's over here between these monomers. They have a distinct name for each one of these macromolecules. So just like before, we're going to stitch together a bunch of videos so you can take this with your notes. And then what I'd like to see you do, and we do have notebook checks. They're pretty big grades. They're meant to help you. I would take another color of pen, whatever, and as I watch Becca Delosier talk, or the science music guy, or whoever has got the other little video, if somebody says something and I'm like, oh, why didn't Miss Kay say it that way? That makes a lot more sense. Well, then write it in here. Add on to your notes and let those pieces of the videos help make sense of things. So that's what you should be doing as you go. So we're done now with the macromolecule introduction.
Hi, this is Ms. Delosier, and these are your notes on macromolecules. So the first thing we need to talk about is what a biological macromolecule is, and there are 4 types. So they are carbohydrates, lipids or fats, proteins, and nucleic acids. And so we're going to go ahead and we're going to talk a little bit about the basic concept of macromolecules and how they're used in biology, and then we're going to break those 4 types into separate videos. So I do want to talk about the fact that these carbohydrates, proteins, and nucleic acids are all what we call polymers, whereas lipids are not polymers. And that's important to remember. We've talked quite a bit about polymers and monomers in class. So a polymer is simply a long molecule that's made of similar or identical subunits, which we call monomers. So monomer, let's define that. So I've got my basically it's like a string of beads. So if I have a string of beads, the beads can be different colors, they can be different sizes, but they're all strung together in one long chain. And each of those is going to be held together by a covalent bond, and that's pretty important when we're talking about the biological molecules, that they're being held together by covalent bonds. Each of those beads, though, is going to be a monomer, so that's just going to be a building block or an individual subunit. And so that would just be the individual beads before I connect them. And that's really all we're talking about, a string of beads versus loose beads. That's polymer versus monomer. And so that's true for carbs, proteins, and nucleic acids. So let's talk about how we actually bond them together, because I've got these monomers and they're not covalently bonded to each other, and I need to somehow make them into the polymers, which are covalently bonded to each other. So I'm going to build or break down my macromolecules through one of 2 processes, and these should be familiar to you from chemistry. The first that we're going to cover is dehydration synthesis. Dehydration, synthesis. Synthesis means I'm building, and I'm building by removing water specifically. So I'm going to take a monomer, and then I'm going to take another monomer. And if you look at these monomers, when we're looking at these biological molecules, it's very common to have a hydroxide group, an OH, and then a hydrogen on an opposite side. So the way that I'm going to do this is I'm going to remove the hydroxide from one monomer, and then I'm going to remove the hydroxyl group from one monomer, and then the hydrogen from another. So I'm going to take those 2, and that's going to form HOH, which for those of you paying attention is water, and that's going to give me 2 monomers that need to form a bond. So they're going to bond to each other plus HOH. So that's my water, and there is the covalent bond that I've formed, and that's dehydration synthesis. The opposite of that would be adding water to break it down. That's hydrolysis, breaking down with water. Hydrolysis, break down with water. So I'm going to take my monomers, put them together into my polymer that's held together by my covalent bond, and then I'm going to add in some water. And so I've got my polymer, I've got my water, and what's going to happen is that polymer is going to have that HOH go in, and it's going to break that bond. I'm going to get a hydroxyl group on one and the hydrogen on the other, so that hydroxide goes and forms the hydroxyl. The hydrogen goes and attaches to normally a carbon, but not always. It could be a nitrogen. And I am going to get my individual monomers back out. So I've got my 2 monomers. Now this happens over and over and over again. So I could be talking about adding 2 monomers together. I could be talking about adding 2 monomers together and then individually adding like another 50 monomers. So I could have a chain of 51. Or I could be breaking down the whole chain. And these are the basic chemical reactions that we're going to be talking about throughout the entire school year. So it's really important that you understand these 2 processes. If you're having any issues, go ahead and send me an email or come in during tutorials and I'll be happy to help. Hi guys, it's Misty, and I wanted to go ahead and finish up the sorting activity that we started in class today. So you should have picked up the worksheet in class today, and you should basically be done color coding and cutting it. If you're not, you need to do that before you start this video. What we're going to do is we're going to go through the sorting activity together in this video really quick. That way you guys can go ahead and start with the pasting tomorrow in class. So the first thing I want you to do is I want you to just sort by elements. Anything that has only carbon, hydrogen, and oxygen, put in one pile, and anything that has nitrogen, sulfur, phosphorus, I mean carbon, hydrogen, and oxygen too, but if it's got a nitrogen, a sulfur, or a phosphorus, put it in a secondary pile of anything else. So pile 1, just carbon, hydrogen, and oxygen. Pile 2, anything else. Be really careful on the ring structures because some of the ring structures have a nitrogen hidden in them that is not outlined in yellow. So double check your ring structures to make sure that there's not a little nitrogen hiding in the ring. So once you do that, I want to look at the ones that only have the carbon, the hydrogen, and the oxygen. And we're going to just sort those kind of based on how they look. And that's really it, just how they look. So I'm going to make piles, like I've got these long skinny ones with just the green, and then I have these that are just orange and red that are chains but just orange and red. I've got some single ring structures that are pretty much just red. I've got some double ring structures. That one shouldn't have been there. It had a nitrogen. I've got the double ring structures that are just the red, and then I've got some weird structures that have 4 rings. And I'm going to put all the 4 ring ones together, all the long skinny ones with the green together, the long chains of about 6 together, and then the rings that are just red together. And we're going to go ahead and figure out what those are. So these 6 chains I've stacked up, and then I'm taking the rings and stacking them up, and I'm going to show you why in just a second. So let's get rid of the long skinny ones and the 4 ring ones. So if you look at these are my single ring and double ring structures. And if you look at the single ring and double ring structures, you'll notice that those single rings are found in the double ring structures. And then those 6 that I put at the bottom that are basically the same, those are open sugar rings. And if you look online, you can see those. Those are all my carbohydrates. And we'll break those down into the different categories of carbohydrates tomorrow in class. These remaining structures, my 4 fused carbon rings, and then I have these long skinny ones. The 4 fused carbon rings are my steroids. Yeah, steroids. So those are steroids. I gave you 3 examples. The long skinny ones with just the carboxyl group, these are fatty acid chains. And they look really different, but they have the same chemical properties. And so we'll talk about how lipids are similar in class over the next couple days. But these are going to go into the same pile, and that pile is the category of macromolecule that includes steroids and fatty acids, and like I said, that is lipids. So we're going to go ahead, steroids, fatty acids, those are both lipids. So stick those in a second pile. Now once you get that done, we're going to go ahead and we're going to sort everything else that's left, all right? So I'm going to just do this by color. I'm going to take these ones that had the purple and stick them in one pile. Anything that's got a yellow and green together on the, like a yellow on one side and a green on the other side, yellow, green, yellow, green, yellow, green. I'm going to put those all in one. And then these weird ring-shaped structures that have a bunch of orange and red, I'm going to stick those in a third pile. Now some of these orange and red ring structures also have some yellow on them, but they don't have the green. So I'm keeping them kind of separate. And we're actually going to deal with those first. So go ahead and keep those out, put the yellow and green ones off to the side. And you'll notice that these ring structures, there's 5 of them. There's 5 single ring structures, if you look at them. And there's 2 that have a double ring, and there's 3 that are a single ring. And you know what these are. These are guanine, cytosine, adenine, thymine, and uracil, your nitrogenous bases, the bases that make up your DNA. So they're your nitrogenous bases. We can just call them nitrogen bases for short. And then if you look at the ones that had the double ring, these, you'll notice that that top ring is just one of the 5 nitrogen bases. So those are also going to be related. And if you look at the bottom ring structure right there, those are a ribose sugar. So all I did was add a ribose sugar to one of my nitrogen bases, and that makes what we call a nucleotide. Okay, so I'm going to put my nucleotides off to the side, and then I'm going to deal with the weird purple guys. So the weird purple guys, you'll notice, just have 1 phosphate, 2 phosphates, 3 phosphates. What you might not have noticed is that if you take those nucleotides, the nucleotides plus the phosphates equal those purple structures. Those are what we call nucleosides. Nucleosides, okay? So you can see that those structures all kind of build on one another. Those are all examples of nucleic acids, okay? Nucleic acids, DNA and RNA, all right? This last group of molecules we're going to look at is our proteins. It has to be proteins because they're the only one of the macromolecules we have left. And if you notice, on all of them, there's a yellow on the left, green on the right. Yellow on the left, green on the right. Sorry, they're kind of out of focus. Yellow on the left, green on the right, yellow on the left, green on the right. There's a whole bunch of them. And you'll notice the only part that's different is the part that's hanging off the bottom of the center. So I actually want to talk about the structure of these. So I have my amino group on the left, and I know I didn't write it in yellow, get over it. And then on the right, I have my COOH group. So the C double bonded to the oxygen and the OH. That's my carboxyl group. What's different is what's hanging off that bottom, that center area. That's called the functional groups. Those are amino acids. That's it. You should have them all sorted for class tomorrow.
Hi, this is Mr. W from ScienceMusicVideos.com, and this video is about monomers and polymers. Big biological molecules like DNA are built from smaller building blocks called monomers. Let's look at a big molecule that we eat a lot of, starch. Here it is. Notice this repeating unit. It's glucose, and you can see that starch is just one glucose chained to the next over and over again. That makes glucose the monomer, while starch is a polymer, a molecule composed of linked monomers. To remember this, use the prefixes. Mono means one, as in monorail. Poly means many, as in polygon. Think about it in terms of Legos. Individual Lego bricks, those are monomers. The structures you build by putting Legos together, those are polymers. All life is built from a relatively small number of monomers that combine into an astoundingly large number of polymers. Just think of our own bodies with a variety of proteins like muscle, or oxygen-carrying hemoglobin, or thousands of enzymes and millions of antibodies. We build all of these from 20 protein monomers called amino acids. It's just like the alphabet of any language, like English. You can take 26 letters, which are like monomers, and combine them into hundreds of thousands of words, which are like polymers. In cells, protein catalysts called enzymes take monomers and chemically link them together. It's called dehydration synthesis. The enzyme pulls an H, a hydrogen, from one monomer and an OH, a hydroxyl, from the other monomer, and links them together at that point. The H and OH combine to form a molecule of water. That's why it's called dehydration synthesis. Here's a sweet example. Here are 2 carbohydrate monomers, glucose and fructose. Through a dehydration synthesis reaction, they combine to form sucrose, or table sugar. Organisms also need to be able to break polymers apart. It's a big part of what happens during digestion. Enzymes break the bonds between monomers by jamming a water molecule in between them. It's called hydrolysis, splitting with water. Of course, monomers can be further broken down. Later on in this course, we'll look at cellular respiration, in which cells take glucose and break it down all the way to carbon dioxide and water as cells make ATP. In addition, monomers have to be made. Later on, we'll see how during photosynthesis, chloroplasts can take carbon dioxide and water and combine them to form glucose, which in terms of matter and energy is the starting point for just about everything else. So that's it for monomers and polymers. Right now, go to sciencemusicvideos.com where there's a tutorial waiting for you that'll help you master this material and master many other topics in biology. Please subscribe to my website, subscribe to my channel, and if you're part of a class that's not using science music videos, tell your instructor about it, and please encourage them to contact me to talk about setting up a site license. Thank you so much for joining me. I'll see you at the next video, and I'll see you at sciencemusicvideos.com. Thanks. Hey, this is Mr. W. Welcome to episode 3 of Thinking Biologically, the 4 biomolecule families. The molecules that make up living things, biomolecules, fall into 4 families, and 3 of them are right here in this peanut butter and jelly sandwich. The bread and jelly are carbohydrates, family number 1. The peanut butter has fat, which is a representative of family number 2, the lipids. It also has lots of protein, which is our 3rd family. The 4th family is here too, but it's not a food group. It's the nucleic acids, the molecules of heredity. The goal of this video is for you to be able to describe each biomolecule family. Right now, we're just going for the big picture, the overall structure and the biological importance. This video has a follow-up quiz that I strongly suggest you try. You'll see that you'll remember so much more if your learning is active. I also want you to know that I use a bunch of structural formulas in this video. And if you need to, you can learn about how they work in my tutorial and video that's called Covalent Bonds. It has a section about structural formulas. Let's go. Carbohydrates are the first family that we're going to consider. Like all the biomolecules, they're made from building blocks that are chemically combined to create larger molecules. Those building blocks are called monomers, and I have an entire video explaining how these are chemically combined in cells to create larger molecules. Here's one carbohydrate monomer. It's called glucose, C6H12O6. It's made by plants, algae, and photosynthetic bacteria during photosynthesis. Glucose is a simple sugar, or a monosaccharide, and monosaccharides are the monomers of carbohydrates. Put 2 monosaccharides together, you have a disaccharide, like sucrose, also known as table sugar. You like sugar? Me too. That's because we're primates, a group of animals that evolved in trees eating fruits. Primates like us love sugars because they're a quick source of energy, and so do lots of other animals. For long-term storage of carbohydrate energy, plant cells chain together hundreds or thousands of glucose molecules and create starch. It's in plant parts like seeds and roots, and it's found in foods like bread, pasta, potatoes, and rice. Starch is a polysaccharide. Many monosaccharides link together. Another polysaccharide isn't for energy storage, but for structure. It's cellulose, or plant fiber. It makes up plant cell walls, and it's found in foods like lettuce, celery, and other vegetables. Cellulose is also in wood, cotton, and paper, the basic biomaterials of civilization. Let's move on to biomolecule family number 2, the lipids. Lipids include fats like butter, oils like vegetable or olive oil, and waxes. The key characteristic of the members of this family is that they're entirely or mostly nonpolar. Fats and oils are chemically very similar. You can see in the structural formula of a fat that one of their main building blocks is what's called a fatty acid. Fatty acids have a long hydrocarbon chain, and those hydrocarbon chains are loaded with chemical energy. That's why fats and oils have more food energy than any other type of food, 9 calories per gram. That's why bears fatten up for the winter. Fats and oils are also used for insulation, and that's why whales have a lot of fat. Waxes are used for waterproofing. The surface of every leaf is covered with wax. Here's a structural formula of another type of lipid, a phospholipid. These molecules are the key components of cell membranes. Remember how I said that lipids are nonpolar? Phospholipids are different. They have a polar side and a nonpolar side. When phospholipids are placed in water, they spontaneously form structures called bilayers, which are the basis of cell membranes. Another member of the lipid family are steroids. Steroids consist of 4 fused carbon rings with other atoms attached. Steroids comprise some extremely important hormones, such as the masculinizing hormone testosterone and the feminizing hormone estrogen. All right, we're halfway there. If you feel like this is making sense, please leave me a comment. How about something like, "Got it, Mr. W." Proteins make up structures like skin, hair, fingernails, and the lenses of our eyes. Proteins like muscle tissue contract, enabling movement. Proteins called enzymes catalyze and control chemical reactions in living things. Another group of protein action molecules are the antibodies. Antibodies play a key role in our immune system, fighting off infections. In terms of food, meat is mostly muscle protein. That's where you're eating muscle tissue when you eat chicken or beef or fish. Egg white is protein. The chemistry of proteins is complex. For now, all you need to know is that the monomer of proteins are amino acids, and there are 20 of them. The nucleic acids make up the last biomolecule family. DNA, the molecule of heredity, is a nucleic acid. So is RNA. These molecules store and transmit genetic information. The monomers of nucleic acids are nucleotides. One nucleotide, ATP, is life's key molecule for storing and releasing energy and getting work done. You did it! Those are the 4 biomolecule families. To consolidate all of this material in your memory, I want you to do 2 activities on my website. They're really going to help you learn. They're free. You don't need a subscription. One is an interactive concept map where you drag labels into the right spot. The other is a quiz that's a kind of word game. Don't worry about getting the answers right on the first try. With me, you always get another chance. The goal is learning biology